Hey, good evening, everyone. Welcome to the free webinar on DNA barcoding and barcode analysis. This webinar is sponsored by Barcode Biotechnology, India's most affordable DNA sequencing company. All right, we will look into barcoding. What is this barcoding? What is DNA barcode? What is DNA barcode gap? We will look into each and every aspect. And finally, I will introduce few things about the company and its services. So this webinar, I hope, will be more interesting for people from live streams uh, branches and also biosciences. Okay, so we will get into the topic. What is DNA barcoding? So, like a universal product code, like you have seen in the various supermarket products, once the product is synthesized, they synthesize a barcode to store the product information, batch number, how it is synthesized, when it is synthesized, what is an expiry. Everything will be stored in the database system with the binary barcode. It will be given on the product, right? So what is this DNA barcode? DNA barcode is a short fragment of DNA sequences that contains all the information of the biological system. What species it is and do they have a phylogenetic signal? Many more informations are stored in the DNA barcode. Actually, it's not stored. It's the humans. We actually identify a barcode gene, we sequence it, we store them in the database so that if someone have a unknown specimen, using this database support, they can simply identify the species back. That's the intention behind the entire effort. So DNA barcoding is a molecular technique used to identify and classify living organism, biological system based on a short standard region in their DNA. So basically DNA barcoding is all about how to use a DNA sequence to identify an organism. Okay. So why it is needed? Why do we need DNA barcoding? So it is estimated that the Earth may have around 8.7 million species. Homo sapien is one of 8.7 million. And we have documented only 1.5 million. You see, last 200 years of science, here in this graph, it is just, here is a broken line and it is they have reported around 7.8 million. Actually, it is 8.7 now. So we have documented not even 1.5 million out of 8.7 million species. This means thousands of species may have evolved and gone extinct without even being recorded or documented. Okay. So conventional taxonomy do have their limitation. They are very slow. The expertise in conventional taxonomist is actually going down. There are many complexes that lead to this situation where thousands of species are continuously evolved and gone extinct without being documented. Okay. So is it something new using DNA sequence to identify the species? No. For last 45 years, we know from Carl Wu's three kingdom concept, it's been done based on 16S RNA sequences. So especially in microbiology, last 45 years, we've been using DNA sequences to identify microorganisms. It's also little true with plants and animals. But for identifying, if for example, gymnosperms, we've been using ITS sequences for identifying in last 2 decades. In case of different fishes, we are using 28S RNA. So this entire effort about barcoding is to have a uniform gene so that we can know the tree of life. How many species are there, how they are related, how distant they are genetically. All those can be done if you have one gene sequences. So the DNA barcoding effort is all about identifying the biological system. Here is a species. You go for a sequencing, you deposit the barcode in the database. The entire DNA sequencing or barcoding works in this way. Okay. You sample a specimen, sequence the barcode gene, compare it with the database, you will know what barcode it is. Okay. So can we have one gene for all life? Literally not possible. The researchers have proven it is not possible. There is no one common gene between plant and animals. Okay. So for animals, they have declared cytochrome C oxidase 1 as a potential barcode gene. For plant species, they declared RBCL gene and MATK gene. Both two genes are there. MATK usually works better for gymnosperms, angiosperms, And for gymnosperms, RBCL works better. But for many plant species, you need to sequence both the gene for species level identification. And for fungi, we have internal transcript spacer region. And for bacteria, even though we don't call it as a barcoding in microorganism, still this is a taxonomic marker, 6NSRR main gene. Okay, so how does the barcoding works? Let's see. Let's say we have a bamboo species A. First, the conventional taxonomists identify the bamboo species, extract the DNA, do the PCR amplification, and sequence the barcode gene. So those gene, RBCL or MATK gene here, then deposits the sequences in the database, get the accession number, and they, like universal product code, you have a 4-colored barcodes here. And now, when someone who don't know how to identify species A of this bamboo recently you deposited in the database, he can simply go for sequencing the DNA barcode gene. Now the genes are compared with the database sequences and he will instantly know what sequence it is. Okay, so this is a guy, Paul D. Hebert. He's the one initiated this effort of DNA barcoding. He's still active. He's a professor in University of Guelph, Canada. He established International Barcode of Life database. From there, you can see UK Barcode of Life, San Diego Barcode of Life, German Barcode of Life, Indian Barcode of Life, Norwegian Barcode of Life, Polish Barcode of Life, Australian Barcode of Life. There are many consortia that has been initiated around this effort to decode the local biodiversity that they have. Not only geography-wise consortia, even phylum-wise. We have fish bowl, which focuses on both freshwater and marine water fishes. We have marine life barcoding. Census of Marine Life has been barcoding, sponsoring this consortia. We have barcoding of ants. We have consortium of Barcode of Life plant working group. And we have wildlife barcoding, sponge barcoding, Lepidoptera barcoding, all birds barcoding initiative, and many more. Okay. So the entire effort is to decode the local diversity. Why to put an effort to barcode it and store it in database? I will show you why. So yeah, all those things have initiated because of this one paper, okay, which came biological identification through DNA barcodes. This is the paper that has initiated that have fired up the entire scientific community on DNA barcoding. After this paper by 2003, in 2009, the DNA barcodes for land plants. Here, the RBCL and MATK genes were proposed after, inspecting nearly 51 different types of genes for their barcoding capability, and they identified that RBCL and MATK can be excellent barcode for plants. And here in this paper, they have around 2011, they identified that ITS gene, ITS region, not gene, ITS region in the fungal can act as a barcode, which we went on to prove that it is working better for marine fungal identification and discovery. Okay. So locally, I mean in India, the national scenario is, I would say, the first barcoding paper has been published by Annamalai University, Center of Advanced Study in Marine Biology. They got a project from Consortium of Barcode of Life during 2007, and the first paper came around 2009 in Current Science. It's regarding using DNA barcodes for identification of marine fin fishes. Okay, or identifying marine fin fishes using DNA barcodes by Ajmal Khan. So that's the first paper, and we have a Botanical Survey of India next taking up plant barcoding, and Zoological Survey focusing on terrestrial animals. And the National Bureau of Fish Genetic Resources have came up with a couple of good papers in fish barcoding. And Central Marine Research, Fisheries Research Institute, CMFRI, and Center for Marine Living Resources and Ecology in India, they are focusing on deep sea ecosystems, barcoding the deep sea ecosystems. So this is a consolidated list of regional and national barcoding initiatives and taxon-focused barcoding working groups. After creating all these DNA sequences, what they are doing? They are actually depositing these sequences in a dedicated database called Barcode of Life data system. We will call them as a bold database. We have national NCBI and we have EMBL and DDBJ, but still bold system is doing excellent effort. Okay, they dedicatedly collect only DNA barcodes. You can either submit COI sequences, ITS sequences, RBCL and MATK sequences. All these 4 genes they are collecting, and you can also barcode an unknown specimen and compare that sequence in the bowl for accurate identification of your unknown specimen. So such a services are provided here. It is free access. You can check it out. And I would say it's a pride thing when it come to Indian context. Now we have a Indian nucleotide data archive. Like you are submitting sequences to NCBI and EMBL, DDBJ, we also have Indian Biological Data Center, which is providing this service of Indian nucleotide data archive. You can actually submit your sequences and get an accession number. So what is this barcoding and how it works? Okay. So once I have a database full of DNA barcodes, decoding the local diversity. Say I have a barcoded beetle A species and I have deposited the sequence. Now I can sample this unknown beetle and extract the DNA. I can go for PCR amplification, get this chromatogram. I can match the sequence with the database. Now I know the beetle species. I'm not a zoologist. I'm not an entomologist. Okay, I don't know what species it is, but using the DNA barcodes, just by comparing the sequence in the database, I can do it. What is this comparing the sequence in database means? Okay, see, consider this example. Here is a database sequence. Okay, sequence in the sense nucleotide sequence, which is of 4 nucleotide adenine, guanine, thymine, cytosine. So this is my query sequence. I will say this is a reference sequence. Whichever sequence that is already present in the database is a reference sequence, and whichever sequence that is there in the that's a query. That's our unknown specimen from which we have generated this sequence is a query sequence. So we are just matching the sequence with the database sequence. Okay, we are just aligning our sequence with the database. When it is getting 100% match, you can be sure that it is that particular species. So it's very simply put, but there are very complex systems in performing barcoding. For example, to start with, I need a conventional taxonomist help. I need, if I want to produce a database of DNA barcodes, to start with, say I'm working with the Lepidoptera butterfly, I need to have a conventional taxonomist helping me identifying what is the butterfly, documenting the key characteristics of the butterfly species, okay, with multiple photographs, documenting the key characteristics, and then uh, I have to take all the metadata, then subject the same specimen for DNA isolation, PCR amplification, where if it is a butterfly, I will be targeting CO1 gene. So I will be using my CO1 primer here. Even though the entire genome of butterfly is extracted, the PCR, that's where that bottleneck is. We will choose what gene to be amplified here. So we will amplify the target gene here, and then we subject them to DNA sequencing. After sequencing, in the bold, you have to submit your machine output file, you have to submit your metadata, pictures, and everything. Then they will assign something called barcodes. This is like a universal product code. What you are seeing here is a universal product code with 4 colors, each color for each nucleotide, adenine, guanine, thymine, cytosine. Okay. This has nothing to do with the identification. The identification happens when unknown specimen sequence is actually given in the database and is being aligned with the database sequence to find the species. So the question is, like I said, it is a multi-skill level task, DNA barcoding. You need to be a taxonomic expert to identify the specimen. You need to have a molecular biology skills to extract the DNA, run the PCR, and go for sequencing. You need to be a bioinformatician to analyze the machine output file, merge the forward reverse sequences, take out the refined sequences, go for blast analysis, go for pairwise distance data building or phylogenetic analysis to confirm the species identity. Once you are done that, you need to know how to manage the database. You need to be a data analyst who can deposit the sequence, get the accession number, manage the database size and its workflow. Okay. So barcoding really require a multi-dimensional skill set. Okay. See, when you extract a DNA from any biological system, the entire genome has been extracted. It may have 50,000 genes. Can all gene act as a barcode? No. So what is the natural question is, what is the features of a gene to be considered as a barcode gene? Okay, what makes a gene to identify a species, or what make a pathway of choosing one gene which can act as a molecular taxonomical marker? So the first and foremost character of the gene should be low variation within species and high variation between species. What is this variation? Okay. See, the intra-species variation should be always low when compared to interspecies variation. Example, take this Orisa sativa or Orisa barthi, two different species. It's a rice plant, right? Two different species of the genera Orisa. You guys know the hierarchy like this, right? Many species will be there in one single genus. Many genus will be there in single family. Many families are there in an order. Many orders are there in the class. Likewise, the hierarchy proceeds. Okay. When I say low variation, what I meant is when I compare two DNA sequences, say for example, Orisa sativa and Orisa barthi, two sequences of Orisa sativa. Let's start with Orisa sativa alone. The variations, when I say variation, if I spread the 100 base pair DNA strand here, another 100 base pair here, if only one base pair is different in 100 base pair, then I would say two DNA sequences have 1% variation. Because among the 100 base pair is the unit of the DNA sequence. Among the 100 base pair, only one nucleotide is variable. If I have 10 nucleotides varying, I would call them as a 10% variation. Okay, or we can say it is 90% similarity or 10% variation. Okay, when I compare Orisa sativa with Orisa barthi, I may Orisa barthi, I may have 5% variation, meaning 5 nucleotide among 100 nucleotides are variable, meaning they are 95% similar and 5% variable. So this is like a species cutoff. How many base pairs are allowed to be variable among the same species? That's something we need to do. This is something called barcode gap. We will see in later slides. Okay. This is something called deriving a species cutoff. Okay. So for RBCL and MATK gene, 3% is a species cutoff. Maybe so it means 97%, 98%, 99%, 100% similarities are allowed within the species. If it is a different species, there will be 95% or 96% similarity, meaning at least for more than 3% variation. Okay, so this is what variation is variation means. So apart from this one character, that is within the species, the intraspecies variation should be low, and between the species, it should be relatively high. Apart from this, there are other features. For example, cytochrome C oxidase is a mitochondrial gene. Among 12 to 18 mitochondrial genes, you have cytochrome C oxidase subunit 1 gene, which is around 650 base pair. They are actually 1500 base pair in length, but 650 base pair is recognized as a barcode potential region. So they are in high numbers. Mitochondria in the mitochondrial genome per mitochondria is multiple, and there are multiple mitochondria within the cell, meaning the CO1 copies in the organism will be high, can be easily detected in the PCR. That's a point number one. So universal primers are available, meaning I can simply use the mitochondrial CO1 gene of a human or animals, or even any zoological species. I can just take the tissue sample or any samples. I can go for it. This CO1 sequence can recognize the COI and they can go for amplification. After sequencing, we will know what species it is. So universal primer is available. They are short length. They are just 600. Anything less than 800 is okay, because Sanger sequencer have this tag polymerase have this fidelity issue. They cannot amplify continuously for more than 900 base pair or so. So anything shorter is advantageous during PCR. And data availability, because of this huge geographical specific consortia and taxon specific consortia, the database is flooded with many samples. Not enough in my opinion, but still the availability of COI data is huge, so that any unknown species can be identified with ease. Okay, and there are many applications beyond just taxonomy. I will tell you what are those applications. So for plant, this chloroplast genome genes, RBCL and MATK is being used for most of the plant. Both gene sequences are recommended. Okay, so what's happening here is for angiosperms, maturus K gene, the gene that is involved in splicing reaction. After post transcription, this maturus K actually splices, and they involve a active role in for many enzymes involved in photosynthesis. And RBCL, obviously it is around 600 base pair length, and both genes together act as an excellent molecular barcode. In fungi, we will call this ITS as a ITS region, not a gene, because this ITS region starts from the last segment of 18S rRNA, ITS1 gene, 5.8 gene, ITS2 gene. Then we have a initial segment of 28S rRNA. Actually, these are the primer binding sites. Okay, and between them is the ITS region. So that's why we call them as an odd gene, because it's a region consisting of group of genes. Okay. And 16S rRNA for prokaryotes, for both bacteria and archaea, they are the molecular marker. Okay. So what is this species cutoff or barcode gap? All right. I would say it's an excellent topic for bioinformatics research, because even though I said 3% is a species cutoff, anything, 3% variation is allowed in within the species. For many species, there will be a species specific cutoff. For example, even though 97% is a species cutoff for identifying a bacteria, for Salmonella and their groups, you need to have 98% similarity. Okay, only 2% variation is allowed, not 3%. Like, this is how they are deriving this is. They will take the DNA sequences of same species collected from different geography, align it, study the pairwise distance, how many substitutions are taking place within. By doing so, they will come to know among how much percentage of variation is existing. That's how the species cutoff, genus cutoff, likewise, family cutoff, order cutoff, class cutoffs. There are very disproportionate, proportionate cutoffs. That's how the species and genera cutoffs are derived. Okay, why to have such a cutoff? Okay, see, in microbiology, it's a gold standard that if you want to propose a new species, you need to go for sequencing. So when I go for a sequencing, when I do a blast analysis, when I compare my query sequence with the reference sequence, if my sequence percentage of similarity is 96%, instantly I know I have a new species. Okay, because the cutoff range is 3%, it is more than 3%, so which is also not there in the library very clearly. That's why the maximum percentage of identity is 96. So there are other assays like DNA hybridization, whole genome sequencing to declare it as a new species. But for plants and animals, once you give, say for example, I barcode a fish, I did the blast analysis. It says 4% variation, meaning 96% similarity. Which means that fish has been studied for more than 30 years, but no one has barcoded it. Okay, so that is the first time that fish has been barcoded, and it can be submitted. Only when you have such a cutoff values, you can know whether the specimen is already barcoded or not, or it is a new specimen or not. So species cutoff or barcode gaps actually are used to identify a new specimen for first time barcoding, or identify the new species that is being barcoded. So what are the applications of barcoding? Okay, they have a species identification, they have a phylogenetic analysis. They are, because we are using the same gene across the domain now, we can simply compare it with other sequences and try to derive the phylogenetic signal in them. And also in food authentication. For example, if you are buying a tinned canned foods, fish, sardines, preserved, okay, are you sure that each and every species in the can is actually belong to sardines? How to know? DNA barcoding is the way. They are also used in illegal trade monitoring. What is this illegal trade? There are fishing holidays, or there is a restriction of catching the red listed species. But 20 years back, these fishermen, when they are getting uh, dolphins or something, when they take the dolphins in, they know they should not catch, but out of greed or something else, they just cut the dolphins into pieces, store the meat in ice, they will bring back to the store. Sure. The wildlife authority will not have any tools to identify what species it is until now. Now, even a fragment of DNA, a fragment of tissue, 10 milligram is sufficient enough to extract the DNA and know what species it is. So barcodings are actually used in the illegal trade monitoring. They are used to resolve cryptic species like no other time. Conventional taxonomists have a hard time in resolving this. Now barcoding is doing a very good job. Okay, and they are rapid and effective. Okay, entire process uh, you know, can be completed in single day. Okay, they are now used in forensic monitoring, biodiversity monitoring, and many more applications. All right. So this is what I said in disease screening, biomonitoring, even dietary assessment. I can simply evaluate the fecal pellets of animals, and I can simply know what are the diet the animal has consumed. For example, if I can take a fecal pellet of a bat and I extract the DNA from the fecal pellet, go for PCR using RBCL and MATK genes, now I can simply say what are the plant this bat is preferring, the fruits or leaves, whatever they have consumed, what from what plant they are actually consuming. Similar to honeybee, from the honeybee's fecal pellet, we can simply decode what is the plant it is preferring, what kind of flower it is actually preferring. All those things can be decoded using DNA barcoding. So this paper is something amazing. This is actually published by us. Okay, the conventional taxonomist point of view is if there are two different species, they will have two different morphology, right? That's what a general belief system is, right? But see here, during a benthic productivity work along southeast coast of India, this species D and G, when a conventional taxonomist take a look, it's very clear they are two different species, right? I've been working as a molecular biologist to various taxonomic groups. You see here the cilia around the body of this. The cilia are some appendages that are used to absorb the oxygen from the water column. Okay, here we have a protruded cilia, which is completely different from that. But when we barcoded is both shared 100% similarity, meaning both are same species. Then we realized the sampling point is different from the species from where we collected this species. Actually, their oxygen concentration is more than 3 milligram. They are rich in oxygen, but this species is actually collected from oxygen poor environment. Okay, in oxygen minimal zone. So it's a morphological adaptation. Conventional taxonomies, by no way they can distinguish these features. Okay. Barcoding clearly says that even though they are two different morphology, they are of same species because the later one, the species D, is actually doing a morphological adaptation. Okay. So far you have seen what is barcoding, how the barcoding database is built, how anyone, anytime, even non-taxonomist can sample any sample and identify using barcoding. Now I want to introduce you to slowly into something called metabarcoding. Okay, what is this metabarcoding? This is a community barcoding, meaning you don't need to have an individual sample. You can have a group of sample, grind it, extract DNA, go for PCR. Every biological system in that group can be decoded by sequencing, and you can identify them to species level. This is a community barcoding or meta barcoding. Even these days, people working in biofouling, various, they are deploying arms in the coral reef ecosystem. All those, simply the entire community can be scrapped off, DNA can be extracted and can go for community sequence. Okay. So how this, what is the major difference between metabarcoding and barcoding? Metabarcoding, actually individual samples are collected, DNA extracted, PCR and sequencing, and compared to the database. In metabarcoding, community of samples are collected. Okay, not you don't need to have individual samples, still it will work best. Right. So this is not an idea. There are many papers beaming in our community, research community. Will see many papers. They are tracking the adulterated food, complex food using metabarcoding. Okay. So slowly from metabarcoding, I want to take you to the third step of barcoding called eDNA barcoding or environmental DNA barcoding. This is fantastic because now you don't even have to collect the organism for barcoding. You can simply collect a liter of seawater, pass them through a DNA binding membrane, extract the DNA from the membrane, go for PCR and get the amplicon, sequence it. You will know what are the organisms there in the sample. Invasive, non-invasive way of sampling. Okay. See, in eDNA barcoding, simply they will collect the sample, air, water, even soil will do. Go for DNA extraction, target the organism, whichever you want to amplify in PCR. After the PCR, you just go for DNA sequencing and compare the sequences of the database. It will tell you what are the species present in the water. Okay, this is something fantastic because now you can simply decode the entire biodiversity from microorganism to macroorganism that is been in the water. There are many research that have proven that fish DNA that is being excreted from the fishes, Epithelial cell will also come along with the fecal material of the fish and they will find in the aquatic ecosystem. Those DNA are stable for 48 hours. They are resisting microbial degradation and light. It's not only for fishes, even plants will shed their DNA when they grow, right? So the entire aquatic ecosystem or soil or air, they are actually filled with DNA which can be trapped, extracted, PCR amplified. In the PCR, you can decide whether you want to study a bacteria, put a 16S RNA primer. You want to study the plant ecosystem of seaweed, seagrass, mangroves in that patch. You put a RBCL MATK primers. If you want to study the vertebrate, invertebrate, go for COI primers. You can decode entire biodiversity with just 1 liter of water. You can know what has happened in last 48 hours in that ecosystem just by sampling 1 liter of water. Okay. Not only water. Here is an example. Airborne environmental DNA metabarcoding detects more diversity with less sampling effort than traditional plant taxonomy community survey. In plant taxonomy, people used to select the plot. They will go to the plot. They will study each and grasses, forbs, and trees in the plot. But here in this paper, both the things are done. Here they came up with a simple air filtration setup, okay, where they will, they are oriented towards the direction of the air. They are highly dynamic system. They have a filter paper inside. So whatever particles they have collected by filtering the air, from the particle they extracted the DNA, they go in for sequencing, RBCL and MATK gene, and they compare the sequences. So here is a traditional method, this side, and here we have a eDNA method. Okay, you see very clearly eDNA is performing 10 times better than the tradition. The grasses that has been missed in the traditional survey was actually captured in eDNA survey. Similarly, even trees also. So there are lots and lots more fantastic application. Okay, even when the taxonomist is dwelling, we can do very well with the DNA barcoding. We did a similar effort here. We want to study the effect of eDNA barcoding on seaweed biodiversity. So what we did, we went for sampling southeast coast of India. We went for sampling the southeast coast of India, and uh, we collected 1 liter sample in all these 5 sampling stations. Okay. After collecting the samples, we went for eDNA barcoding. To our surprise, there are only 23 species recorded so far in southeast coast of India, 23 seaweed species, but we got 31 species. So we know that the traditional method of survey is missing many things. And also it's very fantastic to see this RBCL gene. There are three groups of seaweeds, red seaweed, brown seaweed, and green seaweeds. You see, this is green seaweed cluster, this is red seaweed, sorry, brown seaweed, and here is a red seaweed cluster. Whatever is marked in the orange is actually sequences we produced via barcoding, and what are the black is the sequences collected from the reference databases. See, BLAST result clearly says it is only 88% similarity, meaning this genera is or family itself is not there in the GenBank. Okay, this is to be barcoded. See here, similarly 90%, 94%. These samples or species are not yet in the GenBank. It can be barcoded for the first time. Okay. It's a very advantageous, and I know many PG students here are aspiring for a publication. This research, barcode gap analysis and first time barcodes are actually easy way to publish. I will show you how. Okay. So even FSSAI and ISO these days are demanding eDNA barcoding for authenticating herbal formulation. Example, in this work you see, when the herbs powder are being prepared, they actually place a DNA binding column over the preparation, whichever is getting evaporated or passing through. Actually, those herbs DNA are bounded there. The company is supposed to preserve this filter paper or column, DNA binding column. From the column, actually, you can extract DNA, go for PCR. You can know what are the herbs. After sequencing, you will know this is a list of herb species they have identified after sequencing by the DNA collected from the formulations. Okay, so these are new technology actually evolving now when you have a DNA sequences produced for them. So this is another paper I was mentioning. Okay, if I have a 1 liter of seawater, you can decode the entire marine ecosystem. Okay, for example, here this is a global sampling effort. Wherever you see a dot, they have sampled 1 liter of seawater and they went for it. So the yellow color actually indicates the total biodiversity. The green color indicates a domain-specific identification of biological system in the marine system, in the marine ecosystem. Blue color indicates the kingdom level, and phylum level is in purple color, and black color is actually targeted assessment. Okay, see, barcode to work well, we need to have a local species individually sampled, sequenced, and deposited in the library. Only when we have a library rich in local barcode species, we can effectively use DNA barcoding. In this work, their identification is only up to phylum level. They did not declare class, order, family, genus, species. Why? Maybe they lack sequences in the barcode database. That's why it's very important. Uh, it's really ironical to see country with poor biodiversity are good in cutting edge technologies. Country like India, rich in biodiversity, we are still yet to progress faster in this direction. Okay. So barcoding is not a myth, it's happening everywhere. Here is a proof. Okay, measuring biodiversity from here and botanical origin authentication of dietary supplements by DNA barcoding approach, and how the marketed plant products are actually authenticated using DNA sequences. Many works are coming up slowly. Okay. So let's talk little business now. Why, if you are a UG students or PG students, I would say it's the right time to get into DNA barcoding because it is a $12.5 billion industry worldwide and it is growing 16.5% annually. It is projected to grow to $40.5 billion by 2032. In India, this progress is even more. It is $1,410 million industry and it is growing by 26.7% annually. So if you are thinking about a job after UG or PG, it's the right time to orient ourselves towards this so that you can reap the market as early as possible. So now I want to give a little introduction about this company, Barcode Biotechnology. Okay, what is this company? This company's objective is to increase the local barcodes or enhance the local database. It is founded in 2013 from little funding from consortium Barcode of Life. It is in Chennai, Hyderabad, Goa, and Bengaluru. The consortia from University of Gulf, Canada actually funded this and it's been functioning for more than 15 years. It's a purely Indian company now. We have been operating and we have produced nearly 10,000 barcodes so far. We have a Megabase 1000 sequencer and ABI sequencer 3700 along with Illumina MySec, producing various giving various barcode services. The company has a PCR workstation and Mermaid 192X synthesizer, primer synthesizer. Okay. They are the actually bread supplier of the company. They give various species identification service. Okay, I would say it's India's most affordable service. I remember submitting my samples and companies are charging 2,000 for DNA extraction, 3,000 for PCR and 4,000 for sequencing. I used to end up spending 7 to 9,000 to sequence one sample. I would say this is India's most affordable service now. They are doing with 1,850 rupees per sample. Any plant sample, animal sample, microbial samples, they can handle it. Okay, they are charging 1,850 for herbal authentication. And even if you give them a mixed culture and tell them what are the cultures you want, they can pick the cultures. They will do the pure culturing. They will do a sequencing, identify your species. They will give back the species as a pure culture. Mushroom identification service using ITS, metabarcoding services, even community samples. Even in the plants, if you have you need to sequence two different genes, multi-locus gene services, forge identification, insect barcoding services, there are many more. Apart from all these services, they are running these academics also where they are teaching students how to handle machine output file, how to do a bioinformatics research, how to identify the barcode gap. Many more are taught in this YouTube channel. Please check it out. So why we need to barcode? So here is a picture, a paper called The Future of DNA Barcoding Reflection from early career researcher. He clearly published, he correlated the number of barcodes so far generated from 2003 to 2021, and see the size of the bubble. The size directly proportional to the number of barcodes being generated. See, in India, the effort is clearly not enough. Okay, so in Indian context, especially for people who are doing PG right now, this is an excellent area to pursue. See, papers can be simply published with one DNA sequencing reaction. See here, first time DNA barcoding of a common shipworm. If you have one DNA barcode, this author has obviously did the blast analysis. He has percentage of 96 or 97. He knows that it is being barcoded for the first time. Now he proposed that as a title and went for it. Okay, so it get me to think, did we India as a rich biodiversity country, we have a lot of endemic species. For example, yesterday I spent 20 minutes in listing out the endemic plant species of India. Okay, I just want to check, because why endemic is, I thought outsiders cannot have a hands on this endemic species. So I listed out few 20 endemic species. I took this Sophora interrupta, I put it in GenBank to see whether they've been barcoded or not. No, there is no data found for Sophora interrupta, not even barcode gene. No gene has been sequenced from Sophora interrupta. So this got me very curious. I did a 20 minutes walk. I listed out all these 12, all these species. These red marked species, plant species, they have no data in the GenBank. Their DNA barcoding is not yet done. So for PG students or PhD students, if you just can, if you are accessible to these barcodes, if you can sequence or deposit, sample this and sequence it, you may end up publishing papers very simple like this. So this is one of my paper. I have nothing to do with isopore taxonomy, but I help the author in sampling, DNA extraction, PCR sequencing. Then I analyze the sequence. I prepare the manuscript. Then while doing the blast analysis, I come to know this is a first time sequenced species. So I gave the title first time barcode of Bathynomus kinsley or something. Then it's just a 25 days work, 5 days of manuscript writing. After communication, it took 25 days to accept. Very decent journal to impact or something they have in regional studies in marine science. That's what I'm saying. If we can identify the species yet to be barcoded in your domain, we can simply go for a simple publications, right? So this is also an objective of Barcode Biotechnology company. They want to do lot of publications. Okay, they want to enhance the local library. That's the intention of running this company. So they are offering the summer internship. They are charging you a little fee. The fees is mentioned here, where they are giving you free sequencing of one sample. That one sample you can decide. It can be a fungi, bacteria, plants, animals, any sample. If you can take such a sample which is yet to be barcoded and take this summer internship, in this internship along with sequencing, they will also tell you how to handle your machine output files, how to merge the files, how to do the blast analysis, how to construct a phylogram. This is a bioinformatics research project. They will indulge you for 14 days. You will know how to barcode and how to analyze the DNA barcodes. Out of this internship, you will also end up publishing one sequences in the GenBank. If it is a first time barcoded, it is a high, likely you can write a manuscript and they will also guide you in publishing the manuscript. So this is an excellent opportunity for students. I would say consider this opportunity. I think June 20 is the last date for application. So check this internship. Okay. I think this is an internship advertisement. All right. So the query number, everything has been mentioned here. You can contact the right person. This is not something they are doing for new. So these are the sequences published by the summer interns before. They have done the sequencing for free for these summer interns. They have published it. So they are also have published a couple of research articles because their barcodes are barcoded for first time. So that's it with the DNA barcoding seminar. I hope we introduced you to the concept of barcoding. We explained what is DNA barcoding and what is DNA barcodes and how this DNA barcoding technology has evolved into metabarcoding technology. And now it is eDNA barcoding technology. But still, we still have the mandate to enhance the local library, hence this summer internship. Okay. So those who are interested, you can be a faculty, postdocs, PhD scholars, MSc students. whichever grade, maybe if you are enthusiastic about molecular taxonomy, if you want to do easy publications, I would recommend this is the area to start. Thank you. Happy barcoding.